Hello, I'm Nathalie Reuter at the University of Bergen, and I would like to tell you about molecular simulations of peripheral membrane proteins. Um, First of all, I'm a computational biologist and also a theoretical chemist by education. And my research group and I, we are very much interested in proteins. Proteins are fairly large molecules. They contain thousands of atoms. And even just the one that I have drawn here on that slide is one of the smallest one we work with. And still it has 2,200 atoms approximately. And then we're also very much interested in cell membranes because we study those proteins that bind and act on cell membranes. So cell membranes are simply the barrier that contains the cell and its component. These are the black lines here. You can see it as a bag that contains the cell inside, so to speak. And there are membranes around the other organelles, the mitochondria, the nucleus, etc., etc. So we're talking about cell membranes, plural, and not only one cell membrane. Those cell membranes, they consist of phospholipids or lipids in general. They're fairly small molecules, at least smaller than proteins. And those cell lipids, those lipids, they can be described as consisting of two parts. The heads, which are made of chemical groups that are polar or charged, and therefore the head is hydrophilic and likes to be in contact of water-like environments. And those molecules, the lipids, they also have tails, which are nothing else than long carbon chains with or without unsaturations, and they are therefore hydrophobic. They really don't like to be in contact with an aqueous environment. So when lipids come together, they will have their tails facing each other, and they will form a bilayer, which is basically what a biological membrane is. Um, those membranes, they are very complex. So they contain many different lipids, which are here just represented by the different colors of the heads. They have shorter or longer tails. They can have no insaturations or one or more insaturations. And we also have small molecules like cholesterol. And then there are also membranes. There are also proteins in those membranes, sorry. And those proteins, they can be transmembrane protein, like the blue one, meaning that they traverse the whole membrane. For example, transporting things from one side of the membrane to the other. And we also have the so-called peripheral membrane proteins, which we are very much interested in. And those are like the green drawing here. They interact with only the surface of the membrane, and they will insert some parts of themselves. So some parts of the protein will be inserted between the lipid tails. So in the hydrophobic core of the membrane, a region which will only accept hydrophobic chemical groups or hydrophobic amino acids. The rest of the protein is exposed to the solvent and fairly hydrophilic. So we are very much interested in what are the driving force taking those soluble proteins to an environment where their hydrophobic regions will be well anchored. So we look at the forces. We sometimes run so-called free energy calculations to understand the binding. And we also look in very much details as what's happening between those chemical groups here, which amino acids interact with which lipids and how. And those proteins, it's clear they need to stay quite a while at the membranes to do their job before they leave again. And we know very little about how long they stay. In the case of that protein, a collaborator of mine, Anne, at the University of Massachusetts in Amherst in the US, has measured or estimated experimentally that this protein stays for about 380 milliseconds at the membrane. This is a very short time. sorry, for our scales, but it's quite challenging for us to study. We use molecular dynamic simulations, which is, I think, the best tool for exactly that type of problems, which actually solve the Newton's equations of motions for each particle of the system. We use a molecular mechanics description of the particles in the molecule so that we can calculate the forces between the different particles. And we use, in general, the CHARM 36 force field. The engine we use for molecular dynamic simulations is most often NAMD2. We also use another one called CHARM based on OpenMM. And typically the integration step for our simulations will be in the order of 1 femtosecond or 2. So if we're going to simulate milliseconds, this is quite a number of integration steps we have to perform. I want to show you two examples very quickly. The first one are Phospholipases D. So those are proteins that are found in the venom of brown spiders. These are not spiders I would like to meet. They are quite venomous. And the Phospholipases D, what they do is they bind to the cell membrane on the cells of their prey, and they will start degrading the lipids there. And that has a number of consequences for the prey, and it really isn't good for it. The thing is that different brown spiders will have slightly different phospholipases D, which will give them the ability to attack different types of lipids. So we were trying to understand what gives this, what are the reasons for that. So we run simulations. Here is just a small summary of it. We run simulations of 3 different enzymes, and that was done by Emmanuel Motusami in my group. And could see, as is known by experiments, by the way, that exactly this enzyme doesn't really like to bind to that type of lipids, which are phosphatidylcholine lipids, PC. But those 2 other enzymes that come from 2 different spiders, they can actually bind to the lipids. So far, so good. We have only reproduced what we know from experiments, but it's always good to see that we can do that only with computer simulations. The interesting thing is that now when we have those simulations, we can go and check in detail what the interactions are, which amino acids on that enzyme and that enzyme allow it to go to these membranes. And we actually found out a number of aromatic amino acids in both enzymes. Sorry, and those amino acids are located at about the same place in each of the enzymes. They make cation pi interactions with the choline head groups of the lipids, and those form a cage, which is not found in the ST enzyme. So it explains why they recognize and can process different lipids. So now when we run these simulations, we have to use high performance computers. They take quite a while. So for these simulations, we used the computer called FRAM. And there we would use about 4.5 days for each of those simulations. Mind you, there is a little bit of pre-processing. We run duplicates or triplicates when we run such simulations, and we have in this study in total probably, I don't know, about 20 simulations. So the simulation time is extremely important. Using Betzy, which we benchmarked for a system of about the same size, this simulation would have been done within 3 days for each of them, with 32 nodes and 32 cores per nodes. With 48 nodes and 64 CPUs per node, we could have less than a day per simulation. So it really accelerates our simulation to use a more powerful HPC. This benchmark were done by Dan, who's a software engineer in my research group. Another example now. The system I just showed you was one of the one of the smallest proteins we're looking at. I want to tell you a little bit about lipid transfer proteins. These are proteins that transfer lipids from one membrane to another one, like here. The use of the protein is that it's going to shield the hydrophobic tails and therefore facilitate the transfer from one membrane to the next. Here is a simulation that was run by Mahmoud Mokadam, who's a postdoctoral researcher in my group. And if you follow the blue lipid, you will see that slowly this lipid is going to insert one of its tails into the protein which is in yellow. We need fairly long simulations to do that. And the length of the simulation is much longer than the previous ones. We would like to have up to a microsecond or even more. A microsecond simulation on FRAM would have taken us 25 days for 1 microsecond. And I remind you that we run many more simulations than the one I'm just showing on the screen. With on Betzy, which is what has been used mostly now for that system over the summer, we can go down to 12, 10, or less than 8 days for only 1 microsecond. So it's a tremendous improvement. And the most important thing here is that we can study larger systems. So to conclude, I would like to say that I've mentioned we run free energy calculations as well, which are slightly more demanding simulations than the one I showed. The importance of this work is that it's helping understanding better the interactions between the proteins and the lipids, and hence it will become easier to target these proteins in drug discovery campaigns. If you weren't convinced already now, CPU is CPU intensive, CPU time is absolutely decisive for this type of activity. And the very important thing here is that now with access to a more powerful supercomputer, we can simulate larger and more realistic systems, more comparable to experiments in the wet lab. And with this, I will thank you for your attention and thank Sigma 2 and the Research Council for funding our research. And I'm